Thanks, Mark, for inviting me. It's great to be here. And so my goal today is to give you a little bit of overview of the mechanisms that could be. We heard a lot about social stressors in the previous talk, and you'll be hearing more about that in the next talk from Dr. Hassanar. And so my goal here is to give you a little bit of an overview of how these systems work, and both at like a hormone level and also some brain mechanisms as well. So we've already heard a lot about different types of social stressors, and we know that these can come from many sources: work, school, negative changes like loss of job, unemployment, divorce, or other types of events like wars or natural disasters. And so we know that these repeated exposures to these types of stressors can actually have adverse effects on health. You'll hear more about that in the next talk as well: cardiovascular disease, metabolic syndrome, and the focus from my lab is kind of more mental illness. So as we heard in the previous talk, a big question people have been trying to answer is, how does stress get under the skin? And so to answer this question, we need to look at the different hormones and neurotransmitters that are released in response to physical and perceived threats. And so there's a lot of different types of perspectives you can think of. So how do these systems originate? Well, some of them at least may have started out responding to threats such as like a predator. You know, if you've ever seen any of Robert Sapolsky's videos, they're really effective and pretty accurate too. And so, you know, they always use the clip of the video of the lion chasing the zebra. Well, right. So today, you know, environmental justice, with a few exceptions, you know, if you're in San Francisco at the wrong time, you're not having a lion or tiger chasing after you. Typically, we deal with more social stressors. And so we think of social stressors as an important source of conflict, both in our daily lives and for others. And it turns out social stressors are very important for animals as well. And so the model I'm going to tell you about a little later focuses on social stress in a rodent model. And it turns out this has been very effective at inducing behavioral phenotypes that we see in humans as well. And finally, humans, we have some of our own unique types of stressors. You know, for those of us that do research and have to write grants to fund our research, grant rejection is a very common form of stressor. So the question then that we'll move on to is, so what are all these hormones and neurotransmitters doing? What's their goal? Well, there's no one goal. They have lots of effects. I'm going to try and use the arrow here as a pointer. Okay, so what you can see in this graph is kind of lumping together all of the stress hormones and sort of asking, what do they do? Well, they do a lot of things: increase heart rate, increase blood sugar levels, increase blood pressure, right? And so if you sort of take a big picture, holistic viewpoint of what are these hormones doing, basically what they're doing is they're making energy available to use now so that you can respond to a challenge, whether that's, you know, a tiger chasing you, or you're getting in an argument with someone, or some kind of other challenge that you're dealing with. What these hormones are doing in a very non-specific way, they're just making energy available. So what I'm going to do here with the rest of my time is I'm going to break up the talk into two parts. The first part's going to focus more on neuroendocrine responses to stress and how this sort of energetic response can generate wear and tear on the body. And I'm going to introduce a term that you'll hear more about called allostasis that's originated from people thinking about animals in their natural environments and is now being applied to human health as well. And then I'm also going to tell you a little bit about research from my lab looking at neurobiological responses to stress. And the reason why I would argue this is interesting is because many of these neurobiological changes that can be induced by stress can last long after the stress conditions have ended. So even if you're able to, say, solve certain environmental justice problems now, the previous exposure is still going to have a long-lasting effect. And we need to understand how that's occurring. Okay, so biological responses to stress. So if you had a physiology class and previously, I'm sure you've heard of the hypothalamic pituitary adrenal axis, but just go over it briefly here. So let's see, got the arrow here. All right, so your brain detects some sort of threat, and the hypothalamus is going to respond. It's going to produce and release a hormone called corticotropin releasing factor. which is what they call it. We're in California, so there's two different, some people like to say corticotropin releasing hormone, but in California we say CRF because that's where it was discovered. Or it's one lab that did a lot of research on this hormone was located in Southern California. So we'll stick with CRF. CRF goes to the pituitary. It induces the release of ACTH, which goes to the adrenal gland, which then stimulates the release of cortisol. Now, cortisol is going to have that effect of making energy available for use. But then it's also going to go up into the brain and pituitary to inhibit its own release. So that's an important aspect of this system is that cortisol exerts negative feedback. It inhibits its own release. And so why is that important? Well, there's been, as I mentioned previously, one person that's been very good about communicating stress research to the public is Robert Sapolsky. And if you want to learn more about some of these things I'm talking about, his book, Why Zebras Don't Get Ulcers, is a really good place to start. It's a pretty good read, and it's a pretty good summary of a lot of the research in this area. And one theme that's come out is that the difference between acute activation of these stress hormone systems versus chronic activation. Acute activation is good, right? We need energy to respond to a threat, and turning these systems on, mobilizing that energy is a good thing. Where we run into start seeing problems is chronic activation of this HPA axis. And this can occur through multiple mechanisms. One is by just repeatedly being exposed to stressors, but another possible mechanism is that the system is ineffective at turning itself off. And so this is where I'm going to introduce this concept of allostasis. So this is, as I mentioned before, this was a concept introduced by John Wingfield at UC Davis and Bruce McEwen at Rockefeller in New York City. And the idea was to think about the energetic cost of responding to stressors. So allostasis literally means achieving stability through change. So the idea is you're using energy to maintain homeostasis. You deal with a threat to maintain your current stable state. And so the term that people have been really looking at a lot and applying to a health context is allostatic load. And that refers to the wear and tear that results from either being exposed to too many stressors or from inefficient management of allostasis. And so the repeated exposure to stressors is pretty intuitive. The more stressors you have to respond to, the more energy that's going to take. That's pretty intuitive. But there's another possible source of allostatic load, and that's inefficient responses. So what you have here is a paper from a really nice review by Bruce McEwen, and it's showing you, let's get this arrow here. So here what you're seeing is the physiological response. So you want to think of that as the energetic cost. And so a stressor begins, the individual has to respond. And what you see is that, right, the hormones are being released, energy is being mobilized, and then, right, the stressor ends. That's what the red line is showing you. And then you have recovery. So that negative feedback that I talked about, right, is kicking in. And so now you have gradual decrease in the levels of those hormones and the energy that's being released, mobilized. Now, if you compare that to this inefficient response on the right side here, where the negative feedback is inhibited. And we know that for certain types of psychiatric disorders, such as depression, that there are large subpopulations of individuals that have ineffective negative feedback. And so what you see here is that after the stressor is ended, we don't have that recovery period. So the lack of negative feedback leads to maintaining the high levels of those hormones, mobilizing energy when you don't need it. And we know that this type of situation is not good for the body. We know that there's various types of evidence, which you'll hear more about later. Increased levels of allostatic load is associated with hypertension, heart disease, obesity, and also, as I mentioned, mental illness. Okay, so as Mark mentioned, my lab uses rodent models. Why do we use rodents? Why do we expose them to stress? The reason is because we can study these neurobiological mechanisms and hormonal mechanisms in a controlled way, and we can understand the mechanisms of what's going on in a way that we just cannot do with correlational work in humans. And so I'm going to tell you about two landmark studies from rodents done by another group that show two critical factors that can help influence the physiological response to stress: control and predictability. And so what you have in these studies is you have two rats, and so they're both going to be exposed to a very annoying electric shock on their tail. Okay, and so here's the important. So they're set up in exactly the same way with one difference. Every time these rats get a shock, this rat on the top is going to see a light. Just before he gets the shock, he's going to see a light coming on. So whereas the other rat doesn't know when the shock is coming. Okay, so the only difference between these two groups is one rat knows when I see the light, I'm going to get this annoying shock, whereas the other rat, it's totally unpredictable. Okay, so that's the difference between these two groups, the predictability of the stressor. Okay, so animal 2 is yoked to animal 1. It gets exactly the same number of shocks. And it turns out when you measure the physiological response, whether it's hormones, blood pressure, the rat that sees the light, that knows when the shock is coming, shows a weaker response. So that's really interesting, I think, and it has, in some ways, it's kind of intuitive, but here with the rat, we can experimentally control the situation and really show that something about predictability dampens down that stress hormone response. Here's another example that I think is pretty fascinating. Again, we've got two rats in a yoked design. They each get exposed to exactly the same number of annoying shocks. The only difference here is the rat on the top can turn a wheel to turn that shock off. When he turns off his own shock, he turns it off for the rat below. But the rat below has no control over what's going on. And so that's what we have here, okay? And so again, when you measure the physiological response, whether you're looking at cortisol or blood pressure, right, the rat on the top that has control over what's going on has a weaker physiological response. And we would infer then that over time would have less allostatic load. So just to summarize this part, stress hormones function primarily to deal with the physical challenges that an individual faces by mobilizing energy. But mobilizing that energy comes at a physiological cost that we can quantify as allostatic load. And so what we find is that consistent stressors that are predictable or controllable generate weaker hormone responses, and then that we infer would be associated with lower allostatic load. So if we're looking for ways to try and reduce physiological responses, making stressors, if you can't eliminate the stressor, making it controllable or more predictable is a way to lower the physiological response. Okay, so now the second part of the talk is going to focus more on neurobiological responses to stress. And so we know that psychological stress can cause long-lasting changes in behavior, and that some of these changes in behavior are linked to changes in gene expression or transcription. And so my lab has been focusing on a model called social defeat stress. And this occurs, social defeat occurs when an individual mouse that you see over here loses an aggressive interaction. And so why is this a useful model? Well, if you have mice or rats living in your backyard, the males actually have territories that they're setting up, and they are trying to get the best spots in the backyard, maybe the spot closest to the compost bin, for example, which is what happens in my backyard in Davis, right? And so competition is something that happens in a natural situation. And so what we can do in the lab is we can set this up in a kind of a controlled and less intense way where we can randomly assign individuals to come out on top or lose these social interactions. And so the reason why we do this is because animals that lose social interactions develop behaviors that resemble aspects of depression and anxiety. And I'm going to show you one behavior in particular, social motivation. And so the way that we do this is we have just 3 very short aggressive experiences where an animal kind of loses to a big bully mouse. And we do this on 3 consecutive days. The mice don't get hurt. There's no injuries or anything, but it is very stressful for the mice during those brief periods. But what's fascinating is that the behavior in the brains of these mice change, and those changes can persist for up to 10 weeks, even though they don't get exposed to any more stress. So they have the 3 bad experiences, that's it. Then they're back into their normal cage with the normal sort of routines of living in the lab, and yet their behavior is still different. And so one of the behaviors that we study a lot is social motivation or social interaction. We know in depression and some forms of anxiety that one important symptom is avoidance of social situations, which actually can make other symptoms even worse. And this is something we can measure really easily in a lab. So the way we do this first is we'll take our test mouse and we put it into a large cage. And what you can see is their natural inclination is to kind of run around and explore it. And this cage that you see on the bottom, right here, right now it's empty. And so you can see it's not really super interesting. Now, watch what happens when we replace that cage with another one that has a mouse inside of it. So you see this, our test mouse here, he's really interested in what's going on in that cage. And we typically do a 3-minute test, and what they'll do is they'll spend, so that's 180 seconds, they'll spend 100 seconds typically within 8 centimeters of that cage. And now we've also done some things to quantify like what they're doing, are they crawling around, sniffing? And so the point is, mice have a very strong drive to investigate new social situations that they're unfamiliar with. And you can see that in this graph here. So I don't have a lot of time, so I'm not going to give you my normal song and dance about what we do, but our lab is unique in the fact that we can study the social stress in males and females. And so what's interesting here is that there's actually we see very few differences in behavior, whether it's the social motivation or other types of behavior that we study. Males and females that have not been stressed, their behavior tends to be very similar. Where we see differences is after they get exposed to the stress. And so for this particular assay, females are more affected. And you can see that, all right, well, it's not showing up particularly well here, the arrow, but you can see that, there we go, that females exposed to stress have reduced social interaction behavior, whereas the males not so much. And you can see that a lot better in this heat map. So where you can see the hot colors here, this is where that little stimulus mouse is, and you can see the males, whether they are control or stressed, are spending a lot of time investigating that stimulus mouse, whereas with the female, you can see there's fewer hot colors here for the stressed female. So we've studied a lot of different systems here today, and I had to I have limited time, so I had to pick what I was going to look at, and I have 5 minutes. So I'm going to cut to the chase. We studied gene expression in the amygdala, the central nucleus of the amygdala. We looked at an enzyme called DNA methyltransferase. DNA methyltransferase adds methyl groups to DNA, and so why is that important? Well, methylated DNA is suppressed. The genes inside that methylated DNA can't be transcribed or expressed. So we think DNA methyltransferase is an important gene that could be regulating long-term changes in behavior. And so the first experiment here looks at control and stress mice. And so again, cutting to the chase, what's interesting is here we see a sex difference in the control animals. Remember, the control animals don't show much of a difference in behavior, but here females have more of this DNA methyltransferase gene. After the stress, though, the female DNA methyltransferase expression decreases to the level of males. We don't know what it's doing yet or why, but until we did this study, no one had ever looked at DNA methyltransferase expression in the central nucleus of the amygdala. So this is, I think we're pretty interested in what this could be doing. Now, the second bit is my one little bit of environmental health. It's a collaboration with the Rosenfeld lab at the University of Missouri, where they looked at bisphenol A. And so you can sort of read the basic design, but here it's a developmental study. So the mice were exposed during gestation and lactation, but not afterwards. And again, we saw that sex difference in DNA methyltransferase, Dnmt1 expression in the control animals, but then the animals that were exposed to BPA, that sex difference went away. And it's really fascinating that both the social and the chemical stressors had very similar effects on this DNA methyltransferase. And so Dr. Rosenfeld and we're trying to write grants, experience a little bit of that grant rejection. We're trying to find ways to sort of follow up on this. What's the significance of this sex difference in DNA methyltransferase? And is there any kind of relationship between the two types of stressors, like a two-hit hypothesis? Does exposure to BPA early in life change how you respond to stress later in life? And so just to sort of quickly summarize, we saw DNA methyltransferase expression was increased in females compared to males. Social stress and BPA had the same effect. It reduced DNA methyltransferase primarily in females. And this is persisting. One other thing, these gene expression analyses were conducted 2 weeks or 2 months after the exposures had ended. So these are long-lasting changes. So just to wrap up, psychological stress has important effects on hormones that regulate energy balance. Factors like control and predictability are important moderators, and psychological stress and endocrine disruptors can alter enzymes that control gene expression, and this may be one factor contributing to the long-lasting behavioral changes that we see in response to these exposures. I just want to finish by thanking the people that helped do the research. Emily Wright, who's in my lab, who wrote up the Dnmt work. She's watching online. Hi, Emily. And then Cheryl Rosenfeld, who was my collaborator from the University of Missouri, who helped us with the BPA studies, or actually was conducting the BPA studies. We did the brain analyses. And then my funding sources from the National Institute of Mental Health. So thanks for your attention.